Hi there, my name is Sydney Shaner and I do undergraduate research under Dr. Amy Williams in the Geology Department. Today we're going to be looking at Qaidam Basin, China as an analog for Gale Crater, Mars, and how we can use our detection method to find organics, and how that can possibly be extrapolated to spaceflight missions. So we look at Qaidam Basin as an analog site because it's hyperarid, which is of course a large characteristic of Mars and very prevalent in studies like these. It also exhibits various geomorphological features that are very, very similar to Mars covered in previous studies, such as yardings, aeolian dunes, fluvial structures, and evaporite playas. So samples were collected from Qaidam, organically cleaned, treated with different internal standards to be found on mass spectra. And in our search for alkanes, alkenes, and fatty acids as our lipid biosignatures, we treated the fatty acid samples with tetramethylammonium hydroxide in order to liberate them more easily in the machine. This converted them to fatty acid methyl esters. These were then run on the gas chromatography mass spectrometer, coupled to a flash pyrolyzer at 600 degrees Celsius, and an example of that spectra can be seen in the method section. Regarding results, when searching for alkanes and alkenes, we did come across a high amount of C26, as you can see by that high peak on the right, which is indicative of a higher plant structure that used to live in these sediments. Regarding fatty acid methyl esters, you can also see great amounts of lipids found in these samples. And there's an even over odd chain length preference. You can see the peaks cropping out on the even numbered chains as opposed to the odd ones, which is consistent with a modern microbial community in these mass spec studies. Because we were able to yield these lipid biosignatures from these sediments, I believe that this is a fruitful method of detection for the possible same organics or others on Mars, and this GCMS and addition of TMSH method can be applied to future spaceflight missions, such as on Curiosity rover, which is currently on Mars with the Sample Analysis at Mars instrument suite. Thank you for listening to my presentation, and feel free to ask any questions. Thank you.